Okay, hello, my name is Rebecca Barrick and I go to Moore Park High School.
I'm Kelly Sue and I go to Temple City High.
I'm Ria Kumar and I go to Northwood High School.
And this is our presentation on how the microbiome and stem cells interact.
So for our abstract, just to summarize it, we chose to focus on the gut microbiome, but specifically on intestinal stem cells. These stem cells are often used to replace epithelial cells, which are the cells that exist in the inner lining of your stomach, when they shed periodically, as well as in case of any injuries. In the past, antibiotics, probiotics, and bacterial strains have been found to impact the gut microbiome, but not much research has been done on the intestinal stem cells themselves. So using our results, we'd like to use the research to help stem cell deficiency in the future.
So the main question that we're trying to answer is, how do probiotics, antibiotics, and bacterial strains that affect the body's microbiome affect stem cell populations? So there has been previous research that shows that antibiotics and similar products affect the microbiome within humans in a negative manner. However, not much research has been done on how the negative effects on the microbiome will affect stem cell populations, since stem cells are very important to the development of humans and the regeneration of cells in general. So stem cells are an essential part of human development. However, there is a limited amount of them. We currently are able to find and cultivate these precious cells, which we are currently using to develop new types of drugs to treat different diseases. We know that the microbiome is negatively affected by antibiotics, which are currently necessary to treat bacterial infections. It is currently under question as to if stem cells are affected by the human microbiome, and if so, how. If stem cells are harmed by antibiotics, which destroy the gut microbiome and can cause dysbiosis, or aided by probiotics, which help promote the gut's natural flora, then this could be indicative that the gut microbiome has an effect on stem cell health. If this is shown likely by the data, this would change how we treat patients with stem cell deficiencies and how we treat diseases in relation to a possible impact on stem cell vitality. So the issue also affects us daily in California. We constantly hear news articles about how restaurants like Chipotle or crops such as lettuce have experienced outbreaks of E. coli, a pathogenic bacteria. If it is found that these pathogens do affect stem cells, we could be having them reduced daily due to these outbreaks.
Currently, researchers are establishing background information by looking at the normal functioning of intestinal stem cells and what they do when they're behaving normally. They normally replace epithelial stem cells when they decompose periodically, as well as when you get injured. There has also been research on cell markers and what they indicate for the intestine. Understanding the function of these stem cells is vital to further carrying out the research on how they can be impacted by the antibiotics, probiotics, and bacterial strains. But another example of how such results have been carried out in a different stem cell location is antibiotics that have been tested with the adipose-derived cells, which are stem cells that exist in your fat. And they have shown that administering penicillin, which normally treats infections like your ear infections, have been shown to negatively impact cell differentiation. This information is vital to guiding research in the intestine and how we can use it to help the future.
So our proposal is to study the effect various conditions in the gut microbiome have as an effect on stem cell production. This would include studying the effect of antibiotics, which are known to greatly reduce the amounts of both healthy and not so healthy bacteria in the gut microbiome. Probiotics, which are intended to restore healthy bacteria, such as lactobacillus, to the microbiome, and also studying the effect of certain pathogenic strains on the microbiome, such as salmonella. Our model organism would be rats, who we've treated with the aforementioned conditions. We would then study their stool samples to assess the bacterial populations within the microbiome, and then extract stem cells from the rats by tagging them with fluorescence proteins. The extracted and tagged stem cells would then be assessed using confocal microscopy to view their abundance and ability to divide. We also could use scanning probe microscopy to examine them, because I found a diagram in a paper discussing how they can sense different types of cells using scanning probe microscopy, and so we could assess the health of the cells based on the information collected by the scanning probe microscopy. So some potential limitations of this project that we may encounter include the large amount of time it would take to test both the effects of antibiotics, probiotics, and pathogenic bacterial strains on rats, and then also having a control group to make sure that our data is viable. Another limitation that we could encounter would include the amount of money and time that would be spent gathering data from the gut microbiome and the stem cells, and then using it to study on the microscope. So discussion. So overall, this project would seek to define the relationship between the gut microbiota and the stem cell health through stimulation of different gut microbiome conditions in rat subjects, and then studying the samples using fluorescence microscopy. A future direction that this project may follow would be looking at treatments for stem cell deficiency and establishing the best conditions for stem cell prosperity. We could also look at different types of microbiomes, so maybe the skin microbiome and how that affects different types of health, or how the gut microbiome affects the nervous system as well, and different stem cell systems.
So these are our contributions, and these are our references, and thank you.
Questions?
Yeah.
Yeah?
How do you plan to get stem cells?
So we focused a lot in the research and the lit review, we saw a lot about intestinal stem cells, and so we would extract the intestinal stem cells from the mice and then look at them underneath the microscopes and judge whether they're being produced a lot, or if like with the bacterial strains, what we would predict would be that they'd be reduced because the bacterial strains might affect that. And so if there's like a decreased amount of stem cell growth, then we could determine how the microbiome affects their growth and production and whether they're healthy and able to divide or not.
So we could also get a sample of where we think stem cells are located, and then we could stain the sample to just single out the stem cell and just cut that sample off and then this stem cell off and let it like propagate.
Other questions? Yeah, Michael?
When you're talking about tagging the cells, are you talking about extracting the cells from the mouse and then trying to use the GFP to identify the stem cells? Or are you talking about trying to put a genetic marker in the cells so that the stem cells naturally glow green and can just be counted?
I was thinking putting in a genetic marker because when we were in the lab with Professor Bentalilo, we saw the microdissection machine, and he mentioned how he used the GFP to be able to extract those from the bones a lot easier. And so that's what we were thinking along with the fluorescence.
So then you could quantitate histology slices by how many green cells are in them.
Yes.
As long as you count enough of them, it should be statistically representative of how many stem cells were present in the tissue.
Yes.
Great.
Another question?
I'll just make one quick comment.
Yeah.
I really like the fact that this is very novel, right? And that you're looking at these connections, but it's very dangerous in science to just go shoot in the dark and say, oh,
Yeah.
these things have no data behind them. So the fact that you're able to find a case like the adipose tissue where you actually have seen an effect like this, you don't want to be, it's great to be the first one to discover something, but it's also very risky territory. It usually comes from observations in other projects that those discoveries are made. And so the idea that you have a defined effect that you know exists, at least for other tissues, I wanted to see if there's an effect on this other tissue. I think it gives you a light in the dark, so to speak, to go ahead and approach this. So I think that's an important aspect when you go search. Otherwise, you have to worry about it being a fishing expedition, is what people would accuse you of in grant proposals.
Mm-hmm. Oh, yeah. Oh, two more questions.
Yeah, please.
Okay.
Yeah, they're in like bone marrow and then adipose
also,
cells.
yeah.
Why are you is there a reason why you're specifically choosing to look at the gut microbiome and the gut stem cells?
So from the pre-reading, we read a lot about the Microbiome Initiative and how it's focusing a lot on the gut microbiome. And when we were doing a literature review, we did find stuff on adipose cells, but we also found a lot on intestinal stem cells. And so that's kind of where it went, because a lot of information, it was like in 2016 that this study was done, and they were defining how epithelial cells interact with stem cells in the intestine. And so that's why our project took that direction.
I was just going to ask, I know you heard from Professor Miller talking about not just what's in the gut microbiota, right, but where it's located. Right? The fact that you can extract out the way that you're talking about now and isolate everything and measure it and quantify it. But the fact that they actually have locations inside the intestine matters, right, and their sort of spatial distribution. So I'm wondering if there was anything you learned this week that might make you think you would be able to do this in a live organism instead of having to extract out the cells. Did you hear anything this week, or is there any possibility you could imagine where the fluorescence technique could be used in a living rat
Oh,
instead of
yeah.
having to isolate the cells from the stool
So
or extract them?
So there are PET scans that can be used. So if we were to, I noticed in the lab that they like injected it through the tail, and they'd have different markers that would attach to different parts. So if they attach to the stem cells, or different, if we wanted to look for different strains of bacteria in the microbiome in the gut, we could see those using the PET CT scans.
Thank you, ladies.
Thank you.
Thanks.